I mean, if you took your cerebral cortex, which is the part of your brain generating the thoughts you're having at this exact second, and you somehow managed to iron it out perfectly flat on a table, it would basically barely be the size of a large dinner napkin.
Right, which is just wild to think about.
It is. And yet scientists and philosophers have spent centuries literally going to war over how that tiny crumpled napkin of tissue actually makes you, well, you.
Yeah, it's a beautifully chaotic piece of engineering because, when you picture the brain, it's really tempting to imagine this neat, color-coded map.
Like the textbook diagrams.
Exactly, like every memory and skill is filed away in its own pastel-colored cabinet. But the reality of the cerebrum is much messier. It's full of hidden valleys and incredibly high-stakes medical anomalies and structural mysteries we're literally still untangling today.
Which is exactly what we're getting into. So welcome to today's deep dive into the seat of thought. Our mission today for you listening is to explore the cerebrum.
We are going to unpack a pretty fascinating stack of research today.
Yeah, we're covering the raw anatomy of how this folded mass of tissue is built, the rather wild history of how early doctors tried to map it, and the real modern medical stakes of understanding the geography inside your skull.
Because the overarching goal here isn't just to memorize an anatomical map. We don't want to do that.
Right.
We're looking at the underlying mechanics.
Right.
We want to explore why the physical structure of your brain dictates everything from your impulse control to, how you learn a physical skill to the way you're processing the sounds of our voices right now.
Okay, let's unpack this. Before we can even look at what the brain actually does, we have to understand how it's physically built to maximize space.
Yeah, let's start with first principles on the hardware. At its most fundamental level, the cerebrum is divided down the middle into two halves, the left and right hemispheres.
But they don't just float independently in your skull.
No, they are bridged by this massive, thick band of tissue called the corpus callosum. You can sort of think of it as a high-speed fiber optic cable transferring data back and forth.
Okay, and that cable is constructed of white matter, right?
Yes, exactly.
Because we hear those terms, white matter and gray matter, thrown around constantly. But in the cerebrum, the spatial arrangement of those two things is very specific. And the colors actually tell us what the tissue is doing.
Right, so the white matter makes up the interior bulk of the cerebrum. It basically acts as the wiring. It consists of bundled axons.
Which are the long extensions of the neurons.
Yeah, the thread-like extensions.
Yeah.
And they are white because they're wrapped in myelin, which is this fatty insulating layer.
Kind of like the rubber insulation around a copper wire.
Exactly like that. The myelin prevents the electrical signal from leaking out, which drastically speeds up the transmission of data across the brain and, down the spinal cord.
But the actual processing, like the heavy lifting of consciousness and calculation, that isn't happening in the interior wiring.
No, it's happening on the very outer surface. That outer shell is the cerebral cortex, and that is made of gray matter.
And that's where the cell bodies are.
Right. Unlike the white wiring, gray matter is densely packed with the actual cell bodies of the neurons. And this highly advanced processing center is shockingly thin. I mean, it's only about 2 to 4 millimeters thick.
Which brings us back to the dinner napkin. All of human consciousness, art, science, all of it is happening in an outer layer roughly the thickness of a few stacked credit cards.
It's incredible. And to maximize that processing power, evolution had to solve a spatial problem. It had to cram as much of that 2-millimeter surface area as possible into a fixed, rigid bone box.
The skull.
Right. And since a flat dinner napkin won't fit into a human skull without crumpling, the cortex folds in on itself.
And those folds create a really specific topography. The ridges of the folds are what anatomists call gyri, and the deep grooves or valleys between them are called sulci.
It is a brilliant way to increase surface area without, needing a larger head.
Yeah, thankfully. And speaking of the sheer volume of processing happening in those folds, the research we're looking at highlights a massive misconception about brain anatomy that stood for decades.
Oh, the neuron count.
Yes. Every pop science article and textbook used to state as absolute fact that the human brain contains 100 billion neurons.
Right, it was a number repeated endlessly.
Yeah.
Mostly because earlier accounting methods were just highly flawed.
How were they doing it before?
Well, historically, scientists would take a human brain, slice it into incredibly thin cross-sections, and then they'd count the neurons in a tiny area under a microscope.
Okay.
And then they just extrapolate that number across the entire volume of the brain. The problem is that neurons are not evenly distributed.
Right. Some areas are dense, some are sparse.
Exactly. Extrapolating from a few slices led to a massive overestimation.
So how did we finally get the real number?
Well, a neuroscientist named Susanna Herculano Husel developed a much more accurate method called stereological counting.
And the technique sounds a bit visceral.
It does. She essentially dissolved brain tissue into a homogenized mixture. It's often affectionately called brain soup.
Wow. Brain soup.
Yeah. By destroying the cell membranes, but keeping the cell nuclei intact, she created this perfectly even, uniform liquid.
Oh, I see. So instead of guessing based on uneven slices, you just count the nuclei in a perfectly mixed sample and do the math.
Yes. And that uniform math revealed the entire human brain actually has closer to 86 billion neurons.
Which is a 14 billion neuron difference.
It's a staggering correction.
That is essentially the entire brain of a baboon wiped off the ledger.
Yeah, it really is. And what is even more surprising is where those neurons live. The cerebral cortex, that gray matter, doing the highest level executive thinking we just talked about, it holds only a minority share.
Really? How many?
Only about 16 billion. The vast majority are packed tightly into other structures, predominantly the cerebellum at the back of the head, which manages movement coordination.
Okay, so returning to that folded napkin of the cortex, we should map out the geography. Conventionally, the cerebrum is divided into 4 main lobes, largely named after the overlying skull bones. So at the front, behind your forehead, is the frontal lobe that handles voluntary motor control. And the very front part of that, the prefrontal cortex, manages executive functions. So planning, impulse control, and our core personality.
And then moving to the top back of the head, you find the parietal lobe. This territory deals heavily with touch, spatial awareness, and your physical navigation through the world.
And down on the sides.
Roughly beneath your temples are the temporal lobes. Those are crucial for processing hearing, forming new memories, and managing emotional responses.
And tucked at the very back of the skull is the occipital lobe, which is almost entirely dedicated to vision. Specifically, it contains V1, or the primary visual cortex, which is the very first area of the brain to receive raw visual data from your retinas.
Right.
But, looking at this map, I find myself questioning how real these divisions actually are. Like, if I'm looking at a physical brain resting on a laboratory table, is there a clear anatomical line drawn between the frontal and parietal lobe? Or did early anatomists just draw arbitrary borders on a map for the sake of convenience?
Well, it's actually a mix of both. The borders are not sharp, natural seams in the tissue, like the borders of a country.
Right.
They are defined by specific deep grooves, those sulci we mentioned earlier. The central sulcus, for instance, is a particularly deep valley that anatomists use to separate the frontal lobe from the parietal lobe.
Okay.
And the lateral sulcus, which is often called the Sylvian fissure, that runs along the side and separates the temporal lobe from the frontal and parietal lobes above it.
Okay, so the brain provides the valleys, but we basically decided those valleys represent international borders.
That is a perfect way to phrase it, yeah. The 4-lobe model is just a convention. In fact, many modern neuroanatomists advocate for recognizing a 5th lobe.
Oh, right, the insula.
Yes. It's a region of cortex buried deep inside the lateral sulcus, completely hidden beneath the other lobes. The insula processes interoception.
Which is your conscious sense of the internal state of your body.
Right, like feeling your own heartbeat or getting a stomachache, along with taste and certain emotions.
And then there's also Paul Broca's limbic lobe from 1878, right? Which is a ring of cortex on the inner surface of the hemisphere that manages emotion and memory. And that completely defies the 4 skull bone model. So the tidy map is just a simplification layered over a wildly complex system.
It really is. Which naturally raises the historical question. If this tissue is so homogeneous and complex, how did we ever figure out which specific piece of real estate performs which function?
Yeah, the systematic study of this anatomy started back in 1664 with an English physician named Thomas Willis. He actually published the most detailed map of the brain's physical structure to date, and he even coined the word neurology.
But he didn't know what it all did, right?
Exactly. He was essentially drawing a map of a foreign continent without knowing what any of the cities actually produced. He had the structure, but zero functional theory.
And when there is a vacuum of knowledge, humans tend to fill it with assumptions.
Oh, definitely.
What's fascinating here is how wrong they got it at first. In 1796, an Austrian physician named Franz Joseph Gall introduced phrenology.
Oh, boy.
Yeah. He proposed that specific traits like combativeness or benevolence lived in highly localized specific organs within the brain.
Now, on the surface, the idea of localized function isn't entirely wrong. I mean, we just discussed how the occipital lobe handles vision.
Right.
But Gall's methodology was absurd.
It was completely backwards. His fatal flaw was the physical mechanism he proposed. Gall claimed that as these specific brain organs were used, they grew in physical size.
Like a muscle.
Exactly. Pushing outward and creating distinct bumps on the human skull. He literally taught that you could read a person's entire intellect and moral character simply by running your hands over the shape of their head.
Which is just, it's like trying to guess what software a computer is running by feeling the plastic bumps on its outer case.
That's a great analogy.
It completely ignores the fact that the skull is a thick, rigid structure. It doesn't just bend because you're thinking really hard about math.
Right. The physical mechanics made absolutely no sense, yet it became wildly popular in the 19th century.
Even though it was completely devoid of empirical evidence.
Yes, and furthermore, it was rapidly weaponized to support racist and sexist pseudoscience.
Right.
People began measuring skulls to prove the biological inferiority of certain groups, basically to justify predetermined social hierarchies. It stands today as the ultimate cautionary tale in neuroscience about pattern matching without actual data.
Yeah, it feels like phrenology was doomed from the start because it was obsessed with looking at the outside of the box. To actually understand what these lobes do, someone had to look inside a broken brain and observe what failed.
And that shift from guessing to evidence-based science finally occurred in 1861 in France, thanks to a physician named Paul Broca.
He's the one who encountered the patient nicknamed Tan, right?
Yes, Louis Victor Leborgne. He went by Tan because Tan was almost the only syllable he was capable of speaking.
But his comprehension was fine.
Exactly. His comprehension of language remained perfectly intact.
So when Leborgne passed away, Broca performed an autopsy, and he discovered a very specific lesion, a targeted area of dead, damaged tissue in the lower back portion of the left frontal lobe.
Which today we call Broca's area.
Right. It provided the first rigorous physical proof that a highly specific cognitive function, like speech production, was localized to a specific set of coordinates in the cortex.
And then, just a few years later, in 1874, Carl Wernicke discovered the inverse problem.
The comprehension side.
Yeah, he observed patients who could produce speech fluently, but the words were just a meaningless word salad.
Wow.
Furthermore, these patients could not comprehend spoken language. Wernicke traced this specific deficit to damage in the upper back part of the temporal lobe, an area now known as Wernicke's area.
So this lesion-based science, studying what is lost when a specific piece of the napkin is destroyed, this paved the way for three of the most famous medical cases in human history.
The foundational cases.
Yeah. Let's start with the most dramatic example of frontal lobe function. Phineas Gage in 1848. Here's where it gets really interesting.
Gage's case is definitely foundational to our understanding of the prefrontal cortex.
So Gage was a 25-year-old railroad foreman working in Vermont, and while tamping explosive powder into a rock outcropping, the powder prematurely detonated.
Right.
A 13-pound, nearly 4-foot-long iron rod shot up through his left cheek, passed behind his left eye, and exited through the top of his skull, taking a substantial portion of his left frontal lobe with it, and miraculously, he survived.
The physical survival is astounding. But the medical significance really lies in the reported aftermath.
Because before the accident, his employers described him as this reliable, shrewd, well-balanced leader.
Yes, but after the destruction of his prefrontal cortex, his doctor noted a profound personality shift. Gage reportedly became impulsive, profane, and entirely unable to follow through on any plans. His friends famously noted he was no longer Gage.
Now, historians do point out that the most extreme accounts of his behavior were often exaggerated decades later. There's actually strong evidence he eventually recovered a degree of social function and even worked as a stagecoach driver in Chile.
Which is a job requiring immense focus and planning.
Right. But even with the historical nuances, the core scientific takeaway remains intact. The frontal lobe is the mechanical seat of our impulse control. It basically acts as the brain's braking system.
Exactly. It proved that the essence of who you are has a physical address. So if a frontal lobe injury changes your personality, what happens if you damage the brain's archiving system?
Yeah, right.
That brings us to our second foundational case, a patient known as HM, or Henry Molaison. In 1953, a surgeon named William Scoville attempted to cure HM of severe, life-threatening epilepsy.
By surgically removing large portions of his medial temporal lobes on both sides.
Yes. Crucially, this included a structure called the hippocampus,
And the surgery successfully reduced the seizures, but it resulted in catastrophic anterograde amnesia.
which means HM lost the ability to form any new, long-term declarative memories. He could not remember new facts or events.
A researcher could literally talk to him for an hour, leave the room for 5 minutes, and upon returning, HM would have absolutely no memory of ever meeting them.
It's tragic. Yet the most profound discovery from HM's case came from a researcher named Brenda Milner.
What did she do?
She had HM perform a motor task where he had to trace a star shape while looking only at his hand's reflection in a mirror.
Oh, that's incredibly difficult.
It's a frustrating, totally counterintuitive skill. HM performed the task multiple times over several days. Now, because of his amnesia, every time Milner handed him the pencil, he claimed he had never seen the task before.
Right, consciously he didn't know.
But his performance steadily improved. He got faster and made fewer errors each day.
That feels like a paradox. Like, how can the brain master a physical skill if the conscious mind has zero record of the practice sessions?
Well, it revealed the underlying mechanism of memory. It proved memory is not a single, unified database.
Ah, I see.
The ability to remember facts, faces, and events, declarative memory, relies on the temporal lobe and the hippocampus. But procedural memory learning, a physical skill like mirror tracing or typing or riding a bicycle, that relies on entirely different neural circuits.
Specifically the basal ganglia and the cerebellum.
Exactly. The basal ganglia functions as the brain's autopilot system for learned physical movements. HM's temporal lobe was gone, so his conscious mind couldn't record the lesson, but his intact basal ganglia recorded the physical repetitions perfectly.
Wow.
Yeah.
Okay, so the third foundational case study shifts focus to the corpus callosum, that white matter cable bridging the hemispheres.
All right.
In the 1960s and 70s, researchers Roger Sperry and Michael Gazzaniga studied patients who had their corpus callosum surgically severed. Again, this was to prevent severe epileptic electrical storms from spreading across the entire brain.
And by severing that connection, the left and right halves of the cerebrum could no longer communicate.
Right.
Sperry and Gazzaniga devised these brilliant experiments where they presented visual information exclusively to one eye, isolating the data to a single hemisphere.
Then what happened?
The results were staggering. They proved that each hemisphere could process information and direct behavior entirely independently. They also confirmed that for the vast majority of people, the left hemisphere dominates language production. This work fundamentally altered our understanding of lateralization. It actually earned Sperry a Nobel Prize in 1981.
But, looking at all of this evidence, Broca's area for speech, Gage's frontal lobe for impulse control, HM's temporal lobe for facts, it makes the brain sound like a machine built of completely independent, isolated parts.
Like a sealed-off assembly line.
Yeah. Does the brain actually operate in these rigid compartments?
You've just hit on the central historical tension in neuroscience. It's the great debate between localization and holism.
Okay.
The localizationists, following Broca and Wernicke, believed functions were tied to specific coordinates. The holists, on the other hand, believed the brain functioned as a unified whole.
Right, and the most famous holist from the sources was Carl Lashley, an American psychologist in the mid-20th century. He trained rats to navigate complex mazes to find food.
Right, and once they memorized the route, he surgically removed varying amounts of their cerebral cortex to see where the maze memory was stored.
And Lashley found something that completely contradicted the localization theory, didn't he?
He did. He discovered that the amount of cortical tissue he removed predicted the rat's memory deficits far better than which specific lobe he removed.
Oh, wow.
Yeah, so removing 10% of the cortex caused mild impairment. Removing 50% caused severe impairment, regardless of whether he took the tissue from the frontal or the parietal lobes.
So Lashley called this mass action and acicopotentiality, arguing that complex memories are essentially smeared across the entire cortex, with every part participating equally.
It set up a massive conflict in the field.
Right.
I mean, how do you reconcile Broca's highly localized speech area with Lashley's maze-running rats relying on the whole brain?
Right.
Modern neuroscience resolves this through what we call a connectomic or network perspective.
It reminds me of a symphony orchestra. If we look at the localization argument, yes, you have specific sections. The woodwinds sit in one area, the brass in another.
Right, so Broca's area is the woodwind section.
Exactly, but the holistic argument is also true, because the actual complex behavior of playing a symphony requires a highly coordinated, distributed network spanning the entire stage. You can't play a symphony with just the woodwinds.
That analogy perfectly captures modern network theory. Recognizing a face or moving your thumb relies heavily on specific territories. but any complex behavior requires distributed networks firing simultaneously.
Like reading. Right.
Yes, consider the act of reading a sentence. You need your occipital lobe to visually process the shapes of the letters. You need your temporal lobe to match those shapes to words and assign meaning.
Okay.
Your frontal lobe is actively synthesizing the context to understand the sentence, while your parietal lobe manages your spatial attention, tracking your eyes perfectly across the line of text.
Wow.
All 4 lobes are firing in a synchronized network just to read a single phrase.
Which is incredible. And understanding this networked reality helps us dismantle several lingering pop culture brain myths from the sources. The most persistent one being the claim that we only use 10% of our brain.
Yeah, modern functional MRI scans prove that is unequivocally false. While you don't use every neuron simultaneously, because that would essentially be a massive seizure imaging, shows that activity is distributed across virtually all regions.
Including all 4 lobes over the course of a normal day.
Exactly. The brain is incredibly energy intensive. Evolution wouldn't maintain 90% of it if it were just dormant tissue waiting to be unlocked.
We also need to abandon the myth that the lobes are functionally siloed. As we saw with the reading example, lobes are just convenient anatomical landmarks. They are not sealed vaults operating in isolation.
And perhaps the most pervasive myth is the left brain versus right brain personality type.
Oh, yeah.
It is a gross misinterpretation of Sperry's split brain research. Pop psychology claims that if you are an accountant, you are logical and left-brained, and if you are an artist, you are creative and right-brained.
Which is nonsense.
Total nonsense. While lateralization exists for specific tasks like language, there is absolutely zero empirical evidence that individuals consistently favor one hemisphere over the other to dictate their personality.
It's basically a fabricated personality test, not neuroanatomy.
Precisely.
So bringing this all together, why does knowing the geography of these 4 lobes and their connecting networks matter to you, the listener, today? Like, what are the stakes?
Well, it matters because this physical map provides the working vocabulary for everyday clinical medicine.
How so?
Imagine a patient arriving in the emergency room exhibiting signs of a stroke. Before the doctors even wheel that patient into a CT scanner, they conduct a rapid neurological exam.
Checking their symptoms.
Right. Is the patient's speech slurred? Do they have paralysis entirely on their right side? Have they lost the left half of their visual field?
And those correspond to locations.
Yes. By mapping those specific behavioral deficits back to the known territories of the frontal, motor, or occipital lobes, doctors can often infer exactly which specific cerebral artery is blocked, which shapes rapid, life-saving intervention.
It also dictates how we manage head trauma in everyday life, particularly in sports. The growing awareness around concussions and CTE, chronic traumatic encephalopathy, is fundamentally a story about the structural vulnerability of the frontal and temporal lobes.
Right. Because of how the brain sits in the skull, those 2 lobes take the brunt of the shearing force during impacts,
Leading to the memory and impulse control issues we see in affected athletes.
And the legal system also relies heavily on this anatomy.
That's true.
When a defendant suffers documented damage to their frontal lobe, defense attorneys will argue that their impulse control and judgment are biologically impaired, even if their core intelligence remains intact.
Because the structure of the brain directly influences our legal definitions of culpability.
Furthermore, as we age, the frontal lobes are typically the regions most susceptible to early cognitive decline.
Which is why executive function tests, checking your ability to plan and sequence actions, are standard medical screening tools for older adults.
Ultimately, the physical terrain of the cerebrum dictates the reality of human health.
To recap our mission today, we explored the incredibly dense folded landscape of the cerebral cortex. We updated our neuron count from 100 billion to 86 billion.
Thanks to Brain Soup.
Yes, thanks to Brain Soup. We mapped the frontal, parietal, temporal, and occipital lobes and traced the wild historical journey from the bumps of phrenology to the true network science revealed by patients like Gage and HM.
But before we sign off, I want to leave you with one final kind of mind-bending concept from the research. It illustrates how differently the brain prioritizes the physical world compared to how we see it.
Okay, I'm ready.
It's a concept called the homunculus. In the mid-20th century, neurosurgeon Wilder Penfield mapped the human motor and sensory cortices by applying tiny electrical shocks to the brains of conscious patients.
And what did he find?
He discovered that the brain draws a literal map of your body across the cortex. But if you were to draw a physical creature based exactly on how much brain real estate is dedicated to each body part, you would not look like yourself at all.
You would look like a highly distorted alien.
You would have gigantic, massive hands, enormous lips, and a huge tongue. but a tiny little torso with spindly arms and legs.
Why?
Because the brain doesn't assign space based on physical size. It assigns space based on sensitivity and dexterity. Your fingers require immensely complex neural coordination compared to, say, your thigh. So the brain dedicates vast tracts of the cortex entirely to your hands.
The next time you perform a seemingly simple action, typing a text message, playing a chord on a guitar, or just shaping your lips to speak a sentence, think about that homunculus.
It's amazing to picture.
It really is. Think about the massive folded tracts of your cerebrum dedicating billions of cells entirely to the microscopic movements you are executing effortlessly. The map may be messy, but the engineering is flawless. Thank you for joining us on this deep dive into the seat of thought. Keep questioning the world around you.